Alright, so here we are going to talk about Lecture 4. I'm going to split this into two videos, into Cellular Respiration Part 1 and Photosynthesis. Same set of slides, I'm just splitting them in half. So this first half of the video is probably going to be about 20 or 30 minutes or so, depending on how much I talk. But this is going to talk about cellular respiration first. So let's get into it. So quick overview. Obviously what we're going to cover here is we're going to review all the cell's organelles real quick because again that is probably the most vital and important thing about cell biology. What I want you to get out of this course, it's an intro level course, but cell biology is going to build on your guys' career within medical lab science. Obviously our focus is going to be on the mitochondria specifically in this set of lecture. And students, what I want you guys to do is expected to know, especially the breakdown of glucose to pyruvate when we are discussing glycolysis, the first step of cellular respiration. We're going to look at that picture of glycolysis in detail, and the main things I want you to know and get out of that are the products that are formed and accounting of the ATP production. So when is ATP used, and then how much ATP is produced. Then followed by glycolysis, as you know from the reading. We are then also going to talk about the citric acid cycle. And again from there I want you to also know the products within the cycle and account for how much NADH and FADH2 is made, as well as ATP production. And then ultimately the whole thing ends and is followed up by chemiosmosis or chemiosmosis, which again is the mass production of ATP that occurs in the mitochondria. And remember, that's where we see the flow of hydrogen ions which have been built up in the intermembrane space of the mitochondria, and then they start flowing through the ATP synthase protein channel in the inner membrane of the mitochondria, and then that ultimately makes and pumps out ATP. So that's the main focus of this first half, again on cellular respiration. So again, key organelles that you guys need to be familiar with and know. The nucleus contains the DNA, the master plan that controls cell activities by directing protein synthesis. Ribosomes are cellular components that use the instructions from the nucleus. Again, remember that instructions are written into messenger RNA, and then that messenger RNA gets translated by the ribosomes to build and make the proteins. I can't stress this enough. Review the videos. Any and all videos I'm providing for you guys, definitely review. Not only for each week, but obviously for the exam as well. Then we have the endoplasmic reticulum. Remember, the extensive network of flattened sacs and tubules is a biosynthetic workshop and is composed of a smooth ER and rough ER. And understand what the key difference is between a smooth endoplasmic reticulum versus a rough endoplasmic reticulum. The Golgi apparatus, obviously remember, also contains many transport vesicles that travel within the Golgi. So remember, the Golgi apparatus is also split. You have that cis, medial, and trans Golgi. And as proteins go from the ER to the Golgi, it's within the Golgi apparatus where they are usually modified further. Usually they get sugar groups added to them. These are known as glycoproteins. These glycoproteins get trimmed up and dressed up and ultimately get transported via vesicles, and they're either going to be released out of the cell, say into the bloodstream, like insulin, which is a big major protein that I want you guys to understand and know about, or they could be proteins that are ultimately going to stay within the cell. So again, that's what happens in the Golgi. And then of course our focus today is the mitochondria, which is responsible for carrying out cellular respiration and converting chemical energy of the foods that we eat, and ultimately converting that into ATP, adenosine triphosphate, which is the energy currency that we use within our bodies, that our cells need um, to function and to carry out everyday life. And again, be familiar with the structure, um, obviously, of the mitochondria. Remember that it has a outer membrane, it contains an inner membrane, and then the matrix is the space all the way within, and then of course the cristae are the folds. Okay. Photosynthesis and cellular respiration are very similar processes. Photosynthesis is basically the plant version of cellular respiration. It takes place in the plant cell's chloroplast. It uses energy of the sunlight, is used to rearrange atoms of carbon dioxide and water to produce sugar and oxygen, whereas in cellular respiration, we breathe in the oxygen and we consume the sugar, which is then broken down into carbon dioxide and water. And the cells capture the energy released in ATP, and again, this all takes place in the mitochondria of us as or eukaryotic cells. So again, kind of the circle of life, if you will, at the cellular level. So cellular respiration. So cellular respiration banks energy in the form of ATP. This is again kind of a very simple, straightforward, but very easy to understand chemical formula, if you will, of what goes on. So glucose, remember, is C6H12O6, or it's sugar molecules and sugar um, compounds are usually in this form or within this ratio, and glucose specifically is C6H12O6 and is the fuel that most cells use most often. And basically atoms of the reactant molecules, so reactants are, remember, on this side of the equation are the C6H12O6 and oxygen. Here you have 6 oxygen molecules coming together with the glucose. And then basically this is stored energy, and in cellular respiration we are ultimately going to be releasing energy, and the byproducts of that is going to be releasing carbon dioxide and water. Whereas on the flip side, a plant is going to use the carbon dioxide and water as its reactants, and when it goes through photosynthesis, by using sunlight as its initial form of energy, ultimately it's going to make and generate energy that is going to be stored. So plants basically take carbon dioxide and water, they undergo rearrangement through the process of photosynthesis to release oxygen into our environment and help produce glucose, which is what we as humans and animals consume. And again, vice versa. We use glucose and oxygen as reactants. We go through respiration in order to generate ATP, but release carbon dioxide into the environment and water for the plants. Okay, so understand this. So reactants versus products, and that these two, respiration and photosynthesis, kind of go hand in hand with each other. So now we're going to focus again more on cellular respiration from here on out. So basically what's happening is electrons specifically are being transferred from glucose ultimately to oxygen in order to release the energy in the form of ATP. Oxygen attracts electrons very strongly. So remember this. Remember, there's a word out there for part of your vocabulary from probably chapters 1 or 2, definitely early on, maybe 3. And there's a term called electronegativity. Remember, electronegativity is a number or a value that's designated to an element or a molecule, and it is measuring that the attractivity of electrons to that molecule, so the strength that it attracts. And oxygen happens to have a very high electronegativity value. So that's why oxygen is known as the ultimate electron acceptor, specifically within this process of cellular respiration. So again, cellular respiration is a more controlled descent of electrons. So for example, in the breakdown of during glycolysis from glucose to pyruvate, electrons are being passed on. And remember, so when one molecule or one set of compounds basically lose electrons, that is known as oxidation. And when a substance gains those electrons, that substance is being reduced because it's gaining electrons, and electrons have a negative charge. And we see this process frequently throughout cellular respiration, mainly due to, for example, when NAD plus becomes NADH, that is, the NAD plus is oxidized and is reduced to NADH. And then the electrons get passed on, FAD plus gets reduced to FADH2, and it goes through and on and on. So again, oxidation, you're giving up electrons; reduction, gaining electrons. Oops. Okay, sorry. NADH in the electron transport chain. So again, NAD and as well as FADH as well is played here. These are factors that occur, and so the coenzyme is NAD plus, and again, it accepts electrons and becomes reduced to NADH. NAD plus is an organic molecule that cells make from the vitamin niacin and is used to help shuttle electrons in this redox reaction. So redox is basically reduction in oxidation. So as electrons get passed on, and again, electrons ultimately are going to be shuttled ultimately when glycolysis, remember, happens out in the cytoplasm, but ultimately a bulk of the cellular respiration process occurs in the mitochondria. The electron transport chain specifically are carrier molecules built into the inner membrane of the mitochondria, and it's through a series of these redox reactions that electrons get passed from one carrier to another. So these purple blocks here, shapes, are examples of the electron transport chain. So these are the complexes that make up the electron transport. Notice that they are embedded within the inner mitochondrial membrane. And so this is the matrix, and basically what we are seeing here is a flow of electrons and the constant reduction and oxidation of NAD plus and NADH and FAD to FADH2, etc., all the way down to where oxygen again is our ultimate acceptor of electrons. So all the electrons that go through the electron transport chain ultimately hook up with oxygen, because again, oxygen has a high electronegativity. Okay, so on a scale of like 1 to 10, right, oxygen is a dime piece. All right, if that's how you want to think about it. Everyone wants to end up eventually with oxygen. So that's the electron transport chain, and that's really as detailed as I want you to understand that electrons are flowing, and as electrons flow through the electron transport chain, complexes here, not only are we seeing this constant oxidation and reduction of NAD and NADH and FAD and FADH, but as these processes and as these electrons are going through the electron transport chain, the ETC, you also notice here that we are building a concentration gradient of hydrogens out here in the intermembrane space. And remember, this is important because we want to build a high enough concentration of hydrogens here, because it is this concentration of hydrogens that ultimately are going to power and flow through the ATP synthase where we see chemiosmosis. So again, we're going to see that in detail in just a moment. But that's another important factor of this electron transport chain, is not just that electrons are flowing through and ultimately hooking up with oxygen, but throughout the process, we are building this concentration gradient of hydrogens in the inner membrane space. Again, this is just covering the pathway of the electron transport. So again, most of the chain's components are proteins, which exist in multi-protein complexes. The carriers alternate reduced and oxidized states. So again, as electrons are moving through, obviously certain carriers are giving up electrons, so they're being oxidized. The next set of proteins or substances are accepting those electrons, they are being reduced, and this is a constant cycle. And then electrons drop in free energy as they go down the chain and are finally passed to oxygen, forming water. Okay. Again, just reiterating the facts, again, electrons are transferred from NADH or FADH2 to the electron transport chain. Electrons are passed through a number of proteins, again, the electron transport chain complexes here, including cytochromes, which have an iron atom. Ultimately, again, we're trying to get to oxygen. And the electron transport chain does not generate ATP, but as we will see, it's basically helping to build up to the process of generating ATP. So these are like the three overall main stages, basically, of cellular respiration. So this would be like a summary of cellular respiration. Stage 1, you have glycolysis, occurs in the cytosol, or out in the space within the cell. Glucose is broken in ultimately down into two molecules of a 3-carbon compound known as pyruvate. And then stage 2, that we have the citric acid cycle, which again is now occurring in the mitochondria. So pyruvate is oxidized, and the citric acid cycle occurs and happens within the mitochondria. And that pyruvate is oxidized ultimately to another 2-carbon compound. And again, the main function of these first two stages, glycolysis and the citric acid cycle, is to supply the third stage, the oxidative phosphorylation, with electrons. And ultimately, stage 3, you have again the electron transport chain process is involved, and ultimately we're leading up to chemiosmosis. So again, as NADH and FADH2 help shuttle the electrons to and through the electron transport chain, every time the electrons are passing through those various protein complexes that make up the ETC, we are ultimately going to build up those hydrogens in the intermembrane space, which is ultimately we're going to see help generate the ATP through chemiosmosis. Okay, again, coming up real soon. Alright, so here's the big picture. Okay, these are the things that I want you to really know and really focus on here, specifically for the exam, okay, coming up next week. So I want you to be familiar with this image. I don't want you to worry too much at this point with the structures, but I do want you to know the names and the players involved. So, you know, you could, for example, I'm not saying you couldn't see it, but you might see an image similar to this, and I might give you a few names, and I might want you to fill out the rest. So it's a really crucial process to know and what's happening at each step. So when we start out with glycolysis here, we're starting out with glucose, and we are going to have 2 ATPs being used to energize this process to initiate the breakdown of glucose. And when glucose gets broken down, it's going to first go into a complex, become what's known as fructose 1,6-bisphosphate, which basically means glucose has been converted into a fructose with two phosphates now attached, and those phosphates were obviously donated by the two ATPs, and you have a phosphate group on the first set of carbons and on the second set of carbons, or on the first and the sixth uh, carbons of the fructose. That's where you get the name fructose 1,6-bisphosphate. The 1 and 6 is telling you the location or which number carbon has the phosphate group attached to it. And then ultimately the fructose 1,6-bisphosphate gets broken down further, and it goes through dihydroxyacetone phosphate, which again is going to convert it into G3P, or glyceraldehyde 3-phosphate. G3P is kind of important because not only do we see it here in glycolysis, but we are going to see it later on. So glyceraldehyde 3-phosphate. And then in the second phase of the glycolysis still of stage 1 of the overall process of cellular respiration, what we start to see now is we are going to start generating some things. So here we are going to generate 2-NADH, and basically now we're going to go from G3P to 1,3-bisphosphoglycerate. So again, two phosphates attached at the first and the third carbon. Then again, when we go from 1,3-bisphosphoglycerate to 3-phosphoglycerate, notice we have lost a phosphate because now we are generating an ATP. So that phosphate from the number 1 carbon of the 1,3-bisphosphoglycerate has been lost and hooked up with an ADP, an adenosine diphosphate, in order to make ATP, adenosine triphosphate. So we have some energy being made and released. Now we have 3-phosphoglycerate. We're going to see that we are going to generate some water, and we are going to now become phosphenylpyruvate. And ultimately we're going to go from phosphenylpyruvate to the ultimate product, end product here of glycolysis, pyruvate. And when we go from phosphenylpyruvate to pyruvate, again, same thing. We're going to lose the phosphate, and it's going to be donated to, again, an ADP, and it's going to become ATP. So again, the first half of glycolysis is energy investment. We need 2 ATPs to get the whole thing going. And the second half of glycolysis is energy generation. We're releasing and making ATP at 2 separate points. So what do I want you to get out of this and want you to know? When do we use ATP? When is ATP produced? And the products at each stage. Glucose to fructose 1,6-bisphosphate, fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate to 3-phosphoglycerate, phosphenylpyruvate, pyruvate, and then where amongst those stages do we use ATP, and where within these stages are we making ATP. Okay, biggest things I want you to know and get out of this for glycolysis. Okay. The next stage then we have is the citric acid cycle. So again, this occurs soon after glycolysis. Know that we start with acetyl-CoA, and acetyl-CoA is up here. Now, where did this acetyl-CoA come from? That's something that you guys need to be able to recognize and remember how that happens. So remember, the end product of glycolysis is pyruvate. However, pyruvate is unable to get into the mitochondria where the citric acid cycle, remember, occurs. So there is a small process that occurs that I want you to know and be able to explain from your book, where pyruvate undergoes again a molecular rearrangement, um, and it basically becomes acetyl-CoA, and it is acetyl-CoA that is then able to go into the mitochondria and be the initial product to again jumpstart and go through the citric acid cycle. So basically acetyl-CoA is or was pyruvate. So how does pyruvate go to acetyl-CoA? Know and familiarize yourself with that. As the cycle continues, so this is what I want you to get out of the citric acid cycle. Again, I want you to know that we start out with acetyl-CoA. Again, don't worry about the structures. But I want you to know that once things get going, we're going to go from acetyl-CoA and we go to citrate. Citrate then goes to isocitrate. Isocitrate to alpha-ketoglutarate. Alpha-ketoglutarate to succinyl-CoA, or to succinate. And then succinate to fumarate to malate to oxaloacetate. So know the order of this process. Okay, so we go citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate. That's the order. Then I want you to know where within this process we make NADH and when we make FADH. So we generate NADH between when we go from isocitrate to alpha-ketoglutarate, we generate and make NADH. When we go from alpha-ketoglutarate to succinyl-CoA, we are also making another NADH. And between malate and oxaloacetate, we make NADH. And it's between succinate and fumarate that we make FADH. Okay? So again, these are key things that I want you to get out of the citric acid cycle. Know the order of events, so citrate, isocitrate, alpha-ketoglutarate, etc. So obviously this moves clockwise. And I want you to know where, between what products are we making NADH and FADH. Okay, now we're going to get into chemiosmosis. So remember the whole process then. So we have electrons now that are going to be transferred in through the electron transport chain. And remember, this is going to cause a pump of hydrogen concentration from the mitochondrial matrix to the intermembrane space. And then hydrogens are then ultimately going to move back across the membrane. So remember, we're building up this high concentration of hydrogen ions in the intermembrane space of the mitochondria. And at some point eventually we're going to have so much built up in that intermembrane space that ultimately that concentration is going to be so great that it needs to be released. So the hydrogens will then go through and go down their concentration gradient, and they're going to go through the channel that's built in within the inner membrane, the inner membrane of the mitochondria, known as ATP synthase. So ATP synthase uses this downward flow or exergonic flow of hydrogen to drive the phosphorylation of ATP. This is an example of what chemiosmosis is, the use of energy in a hydrogen gradient to drive cellular work. Okay? And here's how it is. So, again, we have a buildup here of hydrogen ions from what's going on with the electron transport chain. So remember, as electrons are passing through the ETC, every time electrons pass through those complexes of the ETC, we're adding and throwing in hydrogen ions and building up a high concentration gradient up in the intramembrane space. Ultimately, when the concentration gradient is high enough, these hydrogen ions are going to then start flowing down their concentration gradient, and they're going to go through this complex here, which is the ATP synthase. Now, the ATP synthase itself is broken up into two parts. One is called the F0 or F0 region. This is the portion that's within the membrane, so this turbine kind of feature here. And then it has the F1 portion, which is the part here that sticks out into the mitochondrial matrix. And basically what happens is that as hydrogens come through, they're going to go through the F0 or F0 region, and this is going to cause the F0 region to start spinning in a turbine-like fashion, and this causes the whole thing to spin. And it's over here in the mitochondrial matrix where we have a lot of ADP, adenosine diphosphate, and a lot of free phosphates as well. And what happens is as this F1 region of the ATP synthase starts spinning and generating, the ADP and the PI or phosphates hook up and get caught up into this ATP synthase, and as it turns, it's ultimately bringing the ADP and the phosphates closer together and generating ATP. So basically the hydrogens are basically coming through, and they're helping to generate the motion and start pushing the turbine basically of the ATP synthase and start making it start moving. And the ADP and PIs that are free flowing in the mitochondrial matrix get caught up in the ATP synthase, and ATP is generated. That's how it happens. And again, this is where majority of the ATP is generated in cellular respiration. So again, we do have some ATP generated in glycolysis. We do have one or two molecules made of ATP out in during stage 2 citric acid cycle, but it's here at the end through the ATP synthase through chemiosmosis where we generate almost 36 to 38 molecules of ATP. Okay? And it's because of this ultimate process. Okay, and then we have anaerobic harvesting of energy. So we have what's known as fermentation. This is a way of harvesting chemical energy that does not require oxygen. So this provides an anaerobic path for recycling NADH back to NAD+. Be familiar with lactic acid fermentation as well as alcoholic fermentation. Lactic acid is been known, and up to this point is what has been known to, for example, to cause, or what's believed to cause, like the muscle cramps in your body. So a lot of you guys are in the military and you guys train extremely hard. I get that. I know it. I train as well physically. And yeah, your muscles will basically, when you're exerting a lot of force, whether it's in the weight room or your training exercises, you cramp up at times. Well, this is because A, you're not getting enough oxygen when you're training sometimes. So lifting weights, for example, is an anaerobic process. Even though you are told to breathe, sometimes you forget, but you still push through, and you still make that lift happen, but you feel a burning sensation in your muscles. And this, up so far, has been known to be due to a buildup of lactic acid in the muscle cells. But this lactic acid fermentation is still able to produce some energy, even though there's no oxygen. And it's basically the muscle cells can generate NAD+, so NADH is oxidized, so it gives up electrons and becomes NAD+, and this forms pyruvate, which is reduced ultimately to lactate or lactic acid. And the muscle cells can switch to lactic acid fermentation when the cells again need ATP. And this is because, again, there's just not enough oxygen flowing through the bloodstream. So through fermentation, without a lot of oxygen present, we can still generate some ATP. So basically this is what happens when you're trying to push through that last and final rep or that final set. And you feel that burning sensation, but you're told to push harder and harder, but it burns so good. This is what's happening in your body to help make that last rep count, so to speak. And again, I found a phenomenal video done by another professor out there. So check out that video. There's the link there for the YouTube channel, or that video specifically, where he talks about fermentation, what it is, how it occurs. So know that video and be familiar with it, and as well as what the book has told you. Okay? And then alcohol fermentation, used in brewing for beer, winemaking, and baking. And it's where yeast, specifically, recycle their NADH back to NAD plus again, while converting pyruvate this time. Instead of converting pyruvate to lactate, like we saw with the previous fermentation process, pyruvate is converted to carbon dioxide and ethanol. Okay? And again, these are just some examples of organisms that utilize fermentation and what they ultimately generate. So uh, this Propionibacterium, for example, is going to go through a fermentation process, and it's going to release carbon dioxide and propionic acid, and this ultimately makes Swiss cheese. Lactic acid can generate, again, through these types of organisms, so Streptococcus, Lactobacillus, Aspergillus. They, again, also go through the fermentation process. They make lactic acid, which we can see in the end products of cheddar cheese, yogurt, soy sauce. And then, of course, the famous type of yeast here is Saccharomyces, which is used in a lot of experiments as well. But when it goes through fermentation, specifically this type of yeast, it generates carbon dioxide and alcohol, which is used to make the alcohol we consume in the form of wine and beer. So again, just know some of these brief examples of being the end result of fermentation processes. Okay, so that is part one, cellular respiration. Play this video a dozen times, and again, really know the steps of glycolysis, really be familiar with what's happening through the process of cellular respiration, know the citric acid cycle, when and where NADH and FADH and the ATP is produced in that process, and then definitely be familiar with the whole ultimate end product being ATP generation. We ultimately want to make a lot of ATP, but what goes on and what leads up to it, basically that's what cellular respiration is all about. So, glycolysis, citric acid cycle, electrons flowing down the electron transport chain, building up of a hydrogen concentration, ultimately that hydrogen flowing through the ATP synthase, causing the generation of many, many ATP molecules ultimately. Okay? So part two coming up, photosynthesis.